So Josh Knecht is currently a PhD candidate at Binghamton University. And Josh began his academic career at Cornell University, where he worked on the biological control of alfalfa snout beetles. And then after obtaining his BS in plant sciences in 2010, then in 2019, Josh joined the plant breeding and genetics program at Cornell University, where he worked as a technician, researching potato and cereal breeding, she was just talking about. And inspired by this work, Josh joined the lab of Dr. James Sobel at Binghamton University to study the evolution of hybrid incompatibilities at very early stages of reproductive isolation. And Josh uses seedling traits in monkey flowers as the model for this. So with that, I'll let Josh present his talk today, Are Seedling Phenotypes a Source of Reproductive Isolation in Early Divergent Monkey Flowers?
All right, thanks for the intro, Robert, and thank you to SSE for this opportunity to talk about my research with you all. And thank you all for being here. I know this is a really busy time of year for most of us, so I really appreciate you taking the time out of your day to listen to me talk about speciation. So I kind of discovered early on in my undergraduate that what I really liked about biology was plants. And I've kind of taken a winding road to get here, but there was always a tug of wanting to learn more about plants. And so that constant goal, some dumb luck and a lot of help from friends, family, and strangers has led me here to Jay Sobel's lab in the biology department at Binghamton University, where I get to think about how evolution happens in this group of really cool plants that four specimens of which you see on the screen right now. So even though there's pretty clear differences between these plants, they're actually all part of a single species. And while I think the questions of how they became diverged are really interesting, what I'm mostly interested in is what allows them to stay diverged now that they're different. What mechanisms keep these things separate? So using the biological species concept, we think that the process of speciation is the process of restricting gene flow, which would try to homogenize populations. So just really quickly, in order to have gene flow between two divergent populations or species, they have to mate in the first place, produce a zygote that then grows into a fit hybrid. So anything that hinders this process will lead to reproductive isolation between the two groups. And we kind of characterize them in relation to this zygote. So the first big reproductive barriers happen prezygotic. So this is anything that prevents two species from mating with each other, or if they do mate, prevents the gametes from fusing with each other. And a classic example of this in plants is the coevolution of plants with their pollinators. So here is the red and yellow populations of this group that I work in. The red colored plant has evolved to be pollinated by hummingbirds, while the yellow morph is pollinated mostly by hawk moths. And so Sobel and Streisfeld found that pollinator isolation was a good barrier, not complete, but a strong barrier to gene flow in this group. The other class of reproductive isolating barriers happen after. So you have successful mating and fusion of gametes to form this zygote, but for some reason, the hybrids aren't fit. So there's some form of inviability or sterility in the hybrids that prevent them from crossing back to the parents. And there's a really cool example in Arabidopsis where hybrids between, and this has been found in many different strains of Arabidopsis. So hybrids between two parents have misregulations in their immune response, which causes the hybrid to kill itself essentially. And so Christie et al. recently did a meta-analysis looking at across all of plants, mostly angiosperms, but some gymnosperms in there too. And they wanted to see whether prezygotic or postzygotic isolation tended to be stronger. And they found that on average, prezygotic barriers over here to the left tended to be a fair bit stronger than postzygotic barriers over here to the right. And a couple things to think about here. So I think I fall into a trap that's common among scientists, where I study hybrid incompatibilities. So I want to justify why postzygotic barriers might be stronger or more important than prezygotic barriers. But the truth is, they, at least in the initial stages, given enough time, you would probably have a prezygotic or a postzygotic barrier completely shut off gene flow between populations. But during the speciation process, it's going to be a push and pull between prezygotic barriers shutting down migration, and then postzygotic barriers kind of sweeping away any hybrids that get through. Another thing to note here is that in this study, most of the postzygotic incompatibilities were studied in F1 populations. And we'll talk in a minute why I think that might be causing us to underestimate how strong these postzygotic barriers actually are. And so the best model we have, maybe not the best, but a really good model we have for how postzygotic isolation evolves is the Dobzhansky-Miller model. So this invokes negative epistasis between derived alleles. And the big idea here is you start with some ancestral population, and in species one, species one evolves at this A locus, so we have a derived allele at the A locus. Species two evolves at this B locus, so we have a derived allele at the B locus. And so far, both of these derived alleles have evolved together with the ancestral alleles. So there should be no problem between the ancestral and the derived alleles. The problem comes when we make hybrids and you get both an ancestral allele at each locus in a genotype, and this has never been tested in the evolution of these species. And so there's potential here for negative interactions between the A and B locus, which can cause hybrids to be less fit. So I've gotten a lot of use of thinking about these things in terms of a two-locus genotype table. So in this version, the ancestral genotype is this one in the upper left-hand corner. Species one evolves down this left side to have a derived allele. Species two evolves along the upper side, having a derived B allele. And so you have completely fit genotypes on the left and upper side of this table. But these four genotypes in this lower right corner have never been tested, never occurred with each other in the evolution of these two species. And so there's potential here for them to be unfit. And why I think it's important for us to think beyond the F1s is if we're testing F1 hybrid incompatibilities, we're only testing this single genotype. So this double heterozygous genotype. So if this is really unfit, then we can brush off our pants and we're good to go. But just because it's not unfit doesn't mean that these other three genotypes aren't unfit. So we need to look further beyond the F1 to test these other genotypes. And another reason is that we would expect hybrid vigor to be strongest in the F1s. For example, if you have different sets of fixed deleterious recessives in each population, they'll be alleviated in the F1s. This would cause hybrid vigor, which could potentially hide any incompatibilities that happen in the F1s. So this model has been around for quite a long time, but Torelli and Orr formalized it into a mathematical model where they talk about these four genotypes, and they define them in terms of the number of homozygous genotypes involved in the interaction. So in here we have the HO, which is all the genotypes in F1. It's an interaction between two heterozygous loci. Then the H1 genotypes, the H1 genotypes are interactions between a homozygous loci at one locus and a homozygous heterozygous locus at the other one. Then H2 is interactions between two homozygous loci. So in order to estimate the fitness of a hybrid population, we need to know the proportion of these types of incompatibilities and the effect size of these incompatibilities. So thinking about how these might come about, here's a really cool example from monkeyflowers that produce H2 type of incompatibility. So this was found by Zulig and Swigert, where in Mimulus cucutus and Mimulus cutatus, sorry. So what happened is at this first locus, there was a gene duplication, and in cutatus, the original locus became non-functional, and there was never a duplication in Nasutus. So what happens is when you have a homozygous hybrid, a hybrid that is homozygous for the non-functional allele at the first locus, and then has the Nasutus allele at the blank locus, you get these plants these seedlings that completely lack chlorophyll and die. So this is a really strong incompatibility. If a seedling has this genotype, it's completely inviable. So if it's a strong incompatibility, does that translate directly to being strong reproductive isolation? And a lot of people in literature have been using these genotypic landscape plots, and I've kind of taken that up and play with. So we'll talk through that in a minute. So we can define a hybrid genotype in these two dimensions, where on the x-axis here we have ancestry proportion. So this is often called hybrid index. Seems like there's a movement towards ancestry proportion. I will support that. But this is just the probability that an allele at a given locus comes from the parent two. So this axis is defined by parent one on the left, which has no parent two alleles, and parent two on the right, which is all parent two alleles. And then the second axis here is ancestry heterozygosity. This is just the probability that a locus has one allele from one parent and then the second allele from the other parent. And this upper point is defined by the F1s, which are heterozygous for ancestry at all loci. And then, so every single hybrid genotype will fall on this plot. The F2s will be smack dab in the middle here, where they're intermediate for ancestry and heterozygosity, right? We would expect them to have a 1 to 2 to 1 ratio of parents, the heterozygotes, to other parent. And then if you make back crosses between the F1 and the parents, they are right on this the upper legs of this triangle, where they're maxed out for heterozygosity given a certain ancestry proportion. And so even though we might miss, with these 6 crosses, we might miss a broad range of genotypes, especially down here in the lower regions. I'll argue that we can tell a lot about reproductive isolation by what happens in here, especially these upper legs, where we have in order to have gene flow, there needs to be at least a couple of back crosses, right? So if we look at this really strong incompatibility that Zulig and Swigert described, does it translate to low fitness across this hybrid genotypic landscape? And so if we plot the mean fitness of given this phenotype across all the possible hybrid genotypes, you see there's a ridge of high fitness across where the back crosses would be, and even where in the phenotypes where the F2 population would be, the fitness is still the relative fitness is still around 0.95 compared to the parents and the F1s. You get lower fitness down when you have low heterozygosity and intermediate ancestry, so something like a rill, this is where this incompatibility will be most common. So I'll argue that this wouldn't be a very strong source of reproductive isolation. And this may depend on the migration context to some extent, where if you have individuals migrating from one parent population to another, they're always all the hybrids will be up here in the high fitness region. There might be some restriction of gene flow if it's happening through a hybrid zone where there's a lot of individuals in here. But overall, even though it's a really strong incompatibility, it may not translate to strong reproductive isolation. So with the background in hybrid incompatibilities and hybrid fitness, we're going to go look for these things in this Mimulus aurantiacus group, which I showed you a couple pictures of earlier. But it's a really cool group. So there's 2 species in this group. One is Mimulus clevelandii down here at the bottom, and then the rest of these all fall into the Mimulus aurantiacus species complex. So there's 8-ish groups here, and they're spread around central and southern California. So a really nice system for studying speciation during these first steps towards divergence. We have both allopatric, sympatric, and parapatric populations. Red and yellow have a hybrid zone. There's evidence that Calcinus and longiflorus might have hybrid populations. That's still uncertain, but a really cool system for looking at the evolution of reproductive isolation at these first stages. And to date, most of this isolation research has been done in this Panaceus red and yellow ecotype group. And the reason for that is they have this so on the left here, this is San Diego County in Southern California. There's this really nice floral phenotype that we can observe. And Matt Streisfeld has done a lot of work to dissect the genetics of it. But you see this really sharp transition from west to east in San Diego County, where there's red flowers on the west side and yellow flowers towards the east. And if you look at all the chromosomal clines from red ancestry to yellow ancestry, they all follow this sharp cline from west to east. And Stankowski et al. Tried to look at floral QTLs and overlay them and could see if they overlaid with these strongest cline. And for the most part, they weren't able to do that. So there's some indication that there may be reproductive isolation that is not involved with this floral morphology. And then Aidan Short and Matt Streisfeld recently looked at an ancient hybridization in this entire group, and they found a lot of evidence for hybridization throughout the group, but also evidence for selection against gene flow in the group. So we have some clues that there might be widespread, if not like complete resistance to gene flow, there's widespread resistance to gene flow. And so I wanted to ask, are we missing anything? There's no real strong evidence for hybrid incompatibilities. So we might need to look for subtle incompatibilities, and we wanted to see what life stages and which traits might have those incompatibilities. And we came up with a hypothesis that seedlings might be a place in the life cycle where incompatibilities could affect the plant the strongest. So seedlings are under really strong selection, right? I went out into the woods here in New York a couple weeks ago, and there's all these little maple seedlings. So that's what this thing in the red circle is. And I can reach out my hand and cover a dozen or so maple seedlings, and I'm not going to be playing for the NBA anytime soon. So we're talking really dense here, but you know, none of them are going to make it, almost none. Even if we give them a 1% chance, that's maybe wildly optimistic. So really strong selection during this seedling stage. And we see the same thing in our monkey flowers. So on the bottom left here is the seeds from this group. And the ruler, the numbers are showing centimeters. So the little lines there are millimeters. So these are very small seeds. Each fruit can produce hundreds of seeds. And as you see on the right here, each plant can produce dozens of flowers every year. And they're perennial bushy plants, so they can, last a decade or more maybe. So over their lifespan, they can produce thousands, maybe even millions of seeds. And since Southern California is floristically diverse and not monkey flower monoculture, something's happening in this seedling life stage to thin them out. And so we decided to look at seedling size and traits that affect seedling size. There seems to be some evidence that size is involved in recruitment. So this is what we hypothesize that this might be involved. And there's a lot of different things that affect seedling size. Two of the most common talked about are seed traits. So I'm not going to talk a ton about seed traits here, but there's a lot of work being done in monkey flowers and other plants. Some really cool research on how conflict between parental genomes might cause hybrid incompatibilities. So you can see a recent paper by Suleiman Coughlin to talk more about that. What we're really going to talk about today is growth rate and what affects growth rate. And I apologize if there's anybody with a more physiological background here. When I'm talking about growth rate, I'm thinking of the growth in leaf area. I know physiologists typically think about it in biomass. So today I'm talking about leaf area. So the things that affect growth rate, one is allocation. So plants grow fastest when they allocate more resources to photosynthetic capacity. So if they over allocate, they're under allocating to other things. So they may outgrow. If they over allocate to leaves, they may not allocate enough to roots and be susceptible to things like drought. Or if they over allocate to roots or leaves, they may be susceptible. They might not make enough chemical defense and be susceptible to fungal attack or insect attack. So there's a lot of push and pull here that may be the source of negative interactions that can affect these phenotypes. And then the other thing that affects growth rate is just the efficiency that sunlight is turned into photosynthate. So how efficient they photosynthesize and how efficient they are at respiration. So there's potential here for interactions between the chloroplast and mitochondria and the nuclear genome. So overall, I think this is a place where we could find a lot of potential incompatibilities that may have been overlooked, and a lot of cool lines of research if you're interested in different parts of this. There's plenty of things to look at. So to look at this, we're going to use the Calasinus and Longiflorus pair. So that's in this box here, in this phylogeny. So one reason I decided to choose these to look at is their sister to the Panaceous red and yellow, which has had the most work done. And so I thought if we saw any interesting patterns, we can compare to red and yellow and see if there's any parallel reproductive isolation evolution going on in this group. And these guys are from the Southern Sierra Nevada and the transverse ranges here. Take a closer look. So the Calasinus population that we're using comes from the Southern Sierra Nevada, and the Longiflorus is here around LA. And so these guys are spread from the Southern Sierra across the transverse range, with Calasinus getting out to the southwestern Mojave, Joshua Tree area, and Longiflorus in the the western transverse range around Los Angeles. And there's been some reports of hybrids kind of in this area here. My lab mate Rachel and Kruger and I have been kind of trying to look for these things for potential hybrid populations. It's undetermined yet. But you can see they have different floral morphologies. There's a lot of variation in their floral morphology, but they do cluster into two groups. And this is another thing Rachel's looking at, is whether there is pollinator isolation in these groups. We don't know anything about that yet. And then my other lab mate, Irene Martinez, has made F1 crosses between these two to look at seed size and pollen fertility. And we don't have any really clear evidence for post-zygotic isolation from looking at the F1s in those traits. So what I did was made intra parent crosses, so both Longiflorus parental crosses and Calasinus parental crosses, and then I made crosses between them to make an F1 population, and then crosses within the F1s to make an F2 population. So we have both intra-population crosses and then F1s and F2s, and decided to look at these early seedling traits. So the first one is leaf area 1 day after germination. So what I did was germinated them and then took a picture and used ImageJ to measure how big the plants were 1 day after germination. I decided not to use the day they germinated because the cotyledons are still kind of unfurling, and it's unclear how big they actually are. So size 1 day after germination, and then again looking at size 14 days after germination. Same thing, take a picture from above and measured the leaf area. And then I looked at the growth rate between those two time points. And then to see whether anything that was happening at this really early stage, any patterns we saw at this really early stage, were transferred through to the juvenile or adult plants. I measured height at about 4 months after germination. So these are the traits I decided to look at. Now we need a model to connect genotype to phenotype. And I'm a big fan of this line cross model. It's been around for a while, especially with plant geneticists, Mathers and Jinks, and Lynch and Walsh, probably the two best books to dig into it, but it's been used a lot. So it basically takes in genetic effects, so both within and between locus effects, and ancestry. So this is the hybrid, the ancestry proportion and ancestry heterozygosity that we talked about before. And then it goes into this, it's just a linear model, and it predicts the trait mean of a population with a given ancestry. So it's fairly simple, but really versatile. So it's been Demuth and Wade tried to reconcile this with the Torelli and Orr model, and then Fitzpatrick was able to show that it is actually a special case of the Torelli and Orr model is a special case of the line cross model. Rundle and Whitlock, they have added environmental and genotype by environmental effects. So if we want to study how hybrids act in divergent environments, we can do that with this. And then there's been a lot of interest in modeling hybrid incompatibilities using Fisher's geometric model. And Schneemann et al. Was able to reparameterize this line cross model in terms of the genetic effects having geometric interpretations. So it's just a really cool model. We're going to use a pared down version of it today. I don't have enough lines to fit the full model. I only have 4 lines, so I can only fit 4 parameters. So what we're going to look at instead is using it just to test for whether additivity and dominance is an adequate explanation for the phenotypes we see. So if it is an adequate explanation, we would that would be evidence against Dubzhansky-Miller incompatibilities. So remember, the hallmark of Dubzhansky-Miller incompatibilities is this negative epistasis. And so if we have both parental means and an F1 mean, if it's just if the phenotype is just influenced by additivity and dominance, we can predict the expected F2 mean. And Fishman and Willis use this in Mimulus guttatus and Mimulus nasutus crosses to test for whether pollen viability was affected by chromosomal rearrangements or Jablonski-Miller compatibilities. So chromosomal rearrangements would be expected to act through underdominance. So you would be able to see that just with the additivity and dominance model. And they found that their observed F2s were substantially different from their expectation, and concluded that Jablonski-Miller incompatibilities were affecting these phenotypes. And we can look at how this works by just a hypothetical F2 locus that's causative for some phenotype and regressing that phenotype on the number of parent 2 genes. So in this plot, we have on the X, the number of parent 2 genes. So the parent 1 genotype is 0, the F1 genotype is 1, and the parent 2 genotype is 2. And then the line here, so then the dots and the error bars represent each genotype's phenotype. And then the line is the line of best fit. And then these blue dashed lines are going to be the population mean, which in this case is the F2. And remember, in an F2 population, we would expect a quarter of parent 1, 50% parent 2, 50% F1, and a quarter of parent 2. So if we look at a locus that is affecting the phenotype just through additivity, the difference between the parent means and the population mean is the additive component, and they're equal and opposite here. So they'll cancel out. And so if a phenotype is purely additive, the additivity cancels out and we're just left with the F2 mean. If we add dominance into the equation, the additive component is still the difference between the population mean and the regression coefficient, but now we have a dominance deviation that is pulling the means away from this estimate. And so for both the parents, it's going to be negative in this case. So the difference between the estimate and the regression line and the population mean, or the mean for that genotype, will be the deviance, the dominance deviance. And so it'll be negative here for both parents and positive for the F1. And so it's a quarter of this parent, a quarter of this parent, and so 0.5 times negative D, and then 0.5 times positive D, and that just leaves us with the F2 mean again. So if all the genotypic effects are within locus, we can just sum them up across the genome, and we'll just get the F2 mean. So if we look at this, we can compare our observed F2 with our expected F2 to test whether or not there's epistasis. So the next bunch of graphs are going to look similar to this, so I'll just go through this real quick. So on the x-axis, we have the proportion of parent 2, which will be longiflorus in this case. And then on the y-axis will be the phenotypes. And so I've scaled these all to have a value of 1 at the mid-parent mean, so we can compare across phenotypes. And assuming that this y-axis is positively correlated with fitness, if you see an observed F2 that's much lower than the expected F2, that's indicating negative epistasis and potential Jablonski-Miller incompatibilities. And so we'll also plot the difference between the observed F2 and the expected F2. And so negative values of this difference would indicate negative epistasis, potential Jablonski-Miller. So we have a model, we have some phenotypes. Do we see evidence of negative epistasis in these hybrids? So we'll look at the first day after germination first. And you can see, sorry, I forgot to describe these bars. These are just posterior distributions for the mean, with the point being the posterior mean, and then 50% and 95% intervals. So it looks like the observed F2 is much different than the expected F2. And then if we predict from the model what the difference is, the posterior predictive distribution is mostly by a large margin below zero. So really strong evidence that there's negative epistasis here. So the first clue for Dubchansky-Miller compatibilities. And again, these are in terms of the mid-parent mean. So a negative 0.1 here would mean 10% less than the mid-parent mean, the expectation. So for this one day after germination, we have strong evidence for selection or for negative epistasis. I think we have to be a little careful about interpreting this because this is probably largely determined by the seed size, which is at least partially, if not mostly, determined by the maternal genotype. So we need to look out a little further. And if we look at 14 days after germination, this trend gets even stronger. We're now the observed F2 is actually looks like it's outside of everything else. And if you look at the posterior prediction for the difference between the observation and the expectation, you see really strong evidence that there is negative epistasis. Then if we look at relative growth rate, it's a little less clear, but again, the difference between the observed and the expectation is almost entirely below zero. So strong evidence for negative epistasis for growth rate, which I just think is really interesting. So at this early stages, it seems like there's a lot of negative epistasis. If we follow these plants on, do we see that later on? And so measuring height at 4 months after germination, we see that everything, all these lines and expectation are all kind of occupying the same phenotypic space. And the posterior prediction for the difference is also straddling zero. So we don't have any evidence that the additive and dominance model is not a good, it's a fine model for this. So no evidence for negative epistasis here. So taking these all together in these early first 2 weeks of life phenotypes, we see strong evidence for negative epistasis, and then as a juvenile or adult, we see no evidence. So I think we just want to take a second here to highlight that if I had only looked at adult phenotypes, I might have missed this completely. And I think a lot of phenotypes that we do measure in adults. So thinking about where we might find where selection might be acting strongest on organisms might lead us to make predictions about where we can start finding incompatibilities. Yeah. And so my advisor asked me if we could make any predictions about whether about the architecture of these, what kind of interactions do we have? And my initial response was no. But I love playing around with models and just screwing around with them. So I fit a bunch of models that had different combinations of the genetic effects, and I used approximation of leave one out cross-validation to choose the one that had the best out-of-sample prediction. And I projected the relative growth rate phenotype onto our hybrid genotype landscape, and this is what came out. So we have low spot around the back crosses and F2s. So this is indicating that this phenotype might be largely determined by these interactions between homozygous and heterozygous loci. This should be taken with a huge grain of salt. I don't actually have back crosses in this. I do now, so I would like to redo this so I can do this with more confidence. But if this is right, then this is exactly what we'd expect to see for a phenotype contributing to reproductive isolation. So far, we don't have any evidence that, so in order to be involved with reproductive isolation, it needs to hit fitness, right? So far, we don't have any evidence that these traits are involved in fitness. So we looked into the literature, and this meta-analysis by Moles and Westaby found that herbivory, drought, and fungal attack account for 90% of seedling mortality. So we thought we could do something with this, and the most tractable for the greenhouse is drought. So I did a terminal drought experiment to try to tie these phenotypes to some form of fitness. And so this time I measured seed size and germination timing. And again, leaf area 1 day after germination, 14 days after germination, and the growth rate between them. And then when the modal cohort hit 14 days, 14 days old, I stopped watering them and then just counted how long they survived, how many days did they survive. And I had originally hoped that this would stretch out to a week or more, but we had a really hot and dry and sunny spell in the greenhouse, and they died on the 3rd and 4th days after I started the drought treatment. So fitness here is just whether or not they were alive on the 3rd day after the drought treatment. So here's our hypothesized relationships between these phenotypes and survival. So we thought that the size of the plant would be positively correlated with survival. So this could be because bigger seedlings are just more robust, or it could be that size is telling us something about the internal physiology, and if there's problems, that might impact survival, or it could be somewhere in between those two. So we would expect germination day to negatively correlate with size and then survival. Because earlier germinating plants are going to be bigger. Seed size, we would expect bigger seeds to have bigger plants and more survival. And then relative growth rate, higher relative growth rates, we would expect to have larger plants and larger survival. And for the most part, that's what we saw. So looking at the effect of size, or the correlation between size at the start of the drought and survival, we see a strong relationship where bigger plants were just more likely to be alive on that 3rd day. Seed size is a little open-ended. There seems to be a weak positive relationship between seed size and survival, but there's a lot of uncertainty here. One thing I haven't explored is whether there's an interaction between seed size and germination. You might expect this to be more important closer to the day that the plants germinated. Germination day did have a negative relationship with survival. So plants that germinated earlier were bigger and survived longer. And then I just think this is so cool. The relative growth rate had a really strong correlation with survival through size. So plants that grew faster were stronger, were bigger, and survived longer. One thing I should note here is that it's another thing I haven't really played with, but there is a negative correlation between growth rate and germination day, where the plants that have a higher growth rate germinate earlier. So whatever this underlying ambitiousness is, it might extend back before germination. So that's something to look at into the future. So if we think about reproductive isolation in this group, if I had measured it just in the F1, I wouldn't have found any evidence of reproductive isolation. There's no fitness costs in the F1, it seems. But measuring it in the F2, there's a lot of uncertainty about how strong reproductive isolation is, but there's strong evidence that there is reproductive isolation given this test of fitness. So thinking about how we've been testing reproductive isolation, we might be underestimating the postzygotic reproductive isolation. So that's where I am now. What I'm working on currently is doing a similar set of experiments, making the crosses for it now, where I make crosses to this pernicious yellow ecotype, and then I'm making intra-population F1s, F2s, and back crosses between these different phylogenetic distances. So I'm interested to see, are these phenotypes common in this group? And if they are, do they get worse with distance? Does the architecture change with distance? So I'm looking forward to this. And so hoping that you have three big takeaways from this is that I don't think we can evaluate reproductive isolation in single hybrid types. So even F2s, this might give us incomplete understanding of how reproductive isolation is working through hybrids. So I would like to advocate for thinking more holistically about the entire hybrid genotypic landscape. Second is that incompatibility strength may not be proportional to barrier strength. So subtle incompatibilities may lead to real isolation. Uh, I don't know whether these phenotypes I've talked about today are in the wild contributing to isolation. So it's something we would like to do is plant them out and see what happens. So if anybody has a line to the NSF, tell them we could use some money for that. Yeah, so I hope I've convinced you that subtle incompatibilities may make real contributions to barrier strength. And then finally, that these early life history traits could be an understudied source of hybrid incompatibilities. There's lots of lifestyles that plants have, and I've been assuming that fast relative growth rate is better, but in some life histories it might actually be the opposite. And so I'd like to thank my former lab mate Hugger Solomon, my current lab mates Rachel Kruger and Irene Martinez, and my advisor James Sobel. It's been amazing to talk to them and think through these things. And then the undergrads, which helped me out with doing a lot of this work. And then the SSE for the Lewontin Award. It got me to go out to California to actually spend time with these plants, which was amazing. So that's all I have. Thanks for listening.
All right, great. Thank you so much, Josh, for the great talk, making us think about incompatibilities in different areas that we may have not considered before. Uh, if anybody has a question, you can unmute yourself, turn on your camera, or you can type it out in the chat box. I'll give everyone a minute or two to collect themselves. Yeah, so
Yeah.
Susanna has a question that I can read out. Sorry, Josh, you probably just saw it as well. But I'll read it out either way. We'll start with Susanna's question. Do you notice variation in the proportion of seeds that germinate between the parents F1 and F2?
I haven't looked real closely at that, but no, they all seem to germinate pretty well.
So equal across.
Yeah.
Uh,
But again, I don't have a ton of mothers in the crossing design, so I'm a little hesitant to like make any conclusions about that. But for this set of crosses, they seem to do fine. There's other sets that Irene is working on where there's other interesting things going on.
and but that could certainly be another cryptic selective pressure occurring at that stage. Um, And then Carl Nicholas asks, have you looked at the amount of endosperm in seeds? The establishment of seedling is known to be affected by maternal investment, and you briefly alluded to this. There's a large body of literature correlating leaf area, leaf dry mass, and growth rate, even among seedlings. The scaling exponents governing these correlations are particularly informative. I wonder if you might look into these correlations and see if they provide you with some insights.
Yeah, first talking about the endosperm, I'm not looking at that. So that's something that my lab mate here, Irene Martinez, has been playing around with. And there's actually a large group of people in the Mimulus community that are looking at the the especially the conflict between parental genomes and how it affects uh, the endosperm. Um, yeah, in terms of the scaling, I probably should take a closer look at your book to think that through. I have it sitting right here, but uh, thank you.
And so I'll just ask a question. I saw the monkey flowers are distributed on the Channel Island as well, at least one of them. How's that? Since maybe they have even trouble passing barriers on the mainland, yet that one is consistent with the mainland subspecies there on the coast.
Yeah, I can't say too much about that. But actually, someone we're collaborating with, Aiden Short, is working. So he's a grad student in Matt Streisfeld's lab at University of Oregon, and he's looking into that pretty closely. So there's Longiflorus and Parviflorus are on the Channel Islands, and maybe making hybrids. So I think he's got some work he's excited about. So that might be coming out. Sorry, Aiden, if I'm putting pressure. I didn't mean to do that, but he's working on it anyway. Knows a lot more about it than I do.
Gotcha, cool. And Ken Thompson says, okay, I'm going to do a good job with all these words here. Okay. Although it has some strong empirical support from former authors, I often wonder how much the two locus incompatibility model actually reflects common reality versus being a great teaching tool. Okay. The variation in hybrids can tell us something about the genetic architecture of incompatibilities, but it might be difficult to distinguish complex network incompatibilities from many small effect two locus DMIs. Any thoughts on how we can resolve this, or does it matter? Seems like a big question.
Yeah, I don't have a ton of thoughts on how to resolve it. Whether it matters or not, I think it. I think it. The we'd see a lot of similar patterns, uh, given both, right? I don't know if it matters. I think it certainly matters for how we interpret the evolution of them, but in the long term, how it affects reproductive isolation, I don't know if it matters. Yeah, it's a good question. I don't know. That's the answer.
Yeah, he says me neither. So
Yeah.
uh,
Yeah.
it would be nice to solve that here at a Greg seminar series, but uh, and try our best. Um, so if there are any more questions, you can feel free to ask them. I just ask, okay, actually, we got another. Oh, people are thanking you for your talk. Uh, people who support your conclusions. I'm just going to ask in the last minute here, and hopefully you can see those comments, Josh, on your end. Um,
Yeah.
so I don't do any plant work, but the greenhouse is supposed to be stable, right?
Yeah. Yes, it's supposed to be.
Okay.
There's especially cooling it in the summer is a real challenge. So it's susceptible to the really hot, sunny days. We've just had some renovations done, so hopefully that'll be better in the future. But it is influenced by the external environment
Okay.
for sure.
Okay. Gotcha.
It's more stable than than outside, I guess,
Right.
but
Right.
there's still plenty of variation.
But is it worth repeating the experiment since they died so early?
Yeah, so like I said, when I did this, I only had parents F1s and F2s. I now have back crosses as well, so I am going to repeat this to get a better idea of things.
All right, guys. Well, it's 12:59. We're right up against it. So thank you everyone who came here to attend Josh's talk. This was our best attended Greg seminar ever in the 25 or so. I'm not sure exactly how many we've had. So really appreciate everybody's attendance, and we will see you here next week for our final Greg talk of the academic year. Thank you so much, everybody, and thank you, Josh. See you guys.
Thank you, everybody. I really appreciate it. And if you have any thoughts, feel free to send them my way. I'd love to hear
Absolutely.
them.
Reach out to Josh, guys. All right, see ya.